Good afternoon. I'm Dr. Brian Shapiro, a Market Segment Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled Cultivating Diverse Microbes for Laboratory Study and Genetic Engineering and is presented by Dr. Zaira Martin Moldes. Dr. Moldes is a principal scientist for microbiology at Cultivarium. She has a background in biochemistry and biotechnology. She received her PhD in environmental microbiology from the Universidad Autónoma de Madrid, and her research interests include non-model microorganisms, genetic tractability, and high-throughput microbiology. In this presentation, Dr. Moldes will discuss innovative approaches to identify growth conditions and genetic parts in extremophilic bacteria and explore the Cultivarium Portal, a go-to digital resource for non-model microbes. She will highlight the Cultivarium Portal and how you can derive actionable insights for non-model microbes. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. But first, a quick word about ATCC. ATCC is a private, nonprofit organization founded in 1925. We are headquartered in Manassas, Virginia, and also have an R&D and Services Center in Gaithersburg, Maryland. ATCC is a leading global supplier of high-quality biological materials, providing the scientific community with a diverse range of industry-standard products, including microbiome research solutions. We also partner with organizations within government, academia, and industry, which leads us into today's talk. So with that, I'd like to welcome Dr. Zaira Martin Moldes.
Thank you, Brian, for the kind introduction. As you mentioned, my name is Zaira Martin, and I'm a principal scientist for application development at Cultivarium. First of all, I would like to thank ATCC for giving us the opportunity to showcase Cultivarium's work. And today, I'm going to show you how Cultivarium is tackling the availability of information for both non-model and model organisms in this webinar titled Cultivating Diverse Microbes for Laboratory Study and Genetic Engineering. And what is Cultivarium? Cultivarium is a non-profit focused research organization. Our goal is to make open source tools for life scientists to expand the access to new and understudied microorganisms, inspire new research avenues, and push the frontiers of biotechnology. All this by generating and sharing genetic resources and bioengineering techniques. And what is the problem that we are trying to solve? Currently, we are very, very limited to using a small amount of genetically tractable organisms in the lab. But there's a tremendous amount of diversity to access with more than 1 million species of microbes in the biosphere. Essentially, we need to find the right microbe with the right tools for fundamental or basic science applications, such as chemicals and drug production. So to tackle our mission, we are collaborating with ATCC, which is a trusted source of organism collections with more than 11,000 species of microbes to make this extraordinary collection of microbes genetically tractable for scientists. We're very excited to work with ATCC and their extensive catalog so that we can expand the use of non-model organisms to accelerate biomanufacturing and biotechnology research and development. ATCC maintains over 100 years of deposits for their bacteriology and mycology collection that come from diverse and interesting areas, ranging from the human microbiome to extremophilic environments like the National Park Special Collection. And what do we do with the organisms? Cultivarium addresses the most common questions that we hear experimentalists need to know to be able to work with an organism in the lab. How do we grow this organism in the lab? How do we deliver DNA into this organism? How do we genetically engineer its genome? What else is known about this organism? And to answer these fundamental questions, Cultivarium's organism development pipeline is divided in 4 different platforms to tackle each question separately: the culture, delivery, molecular, and data distribution platforms. Now I'm going to show you how we present the answers to these questions to users through our portal. Cultivarium's portal was created as a one-stop shop to get experimental, actionable information for experimentalists about all the microbes that have been banned or described in the world. We currently have more than 140,000 organism pages where we aggregate data and publications for bacteria, fungi, protists, and archaea. Our portal also allows for an easy way to search for this experimental information. We have built a simple search for complex data sets that supports searching for groups of organisms. For example, in this question that I'm showing, we requested growth information for this group of organisms. And what you get is the experimental information in a simple table output. But if you're not sure of what to ask, we have many other example questions that we provide as templates so you can get familiar with this tool. For the purpose of this talk, I'm going to focus on a group of organisms, extremophiles. These are organisms that thrive in extreme environment conditions that include pH, salinity, or radiation. In general, these organisms are not well studied because they are not easy to grow in the lab, and sometimes it's difficult to replicate their native conditions. But they have great potential for many, many applications. And ATCC has many extremophiles to explore in their collection. From ATCC's catalog, we started working with a group of extremophiles: alkaliphiles, radiation-resistant, and halophiles, ranging in applications from alternative bioproduction host to production of industrial enzymes. And to showcase all the information that we have available in the portal, I would like to focus on one organism, Calcalibacterium halodurans, which is highlighted in yellow here. We selected this organism because it's an alkaliphile that is known for the production of alkaline stable enzymes for a wide range of applications like detergent manufacturing, biofuel production, and waste treatment. And when you look up an organism in the portal, the first thing that it shows is the species level information. And next to the species tab, there is a strain tab. When you click on the strain tab, one of the things that we did is to map all the strain sources and reference databases available for each species. All can be found in one place. So you can easily find the ATCC strain link to purchase this strain. In this table, CVM and the numbers below mean that Cultivarium has this strain. Then from this display, you can select the specific strain that you're looking for and what type of information can you find in the strain page. So to start, you can find information on growth. What we do is we determine the optimal growth media, and we test up to 24 media formulations using our media screen. We track the growth over time, and this growth is measured as optical density in a set of common media that we call base media. We then adjust the formulation to the specific growth needs. So for example, we found that the best formulation for this organism is growing in Horikoshi medium 1 that has been tuned to a pH of 10. In here, we also summarize and rank the media that supports the growth for the different organisms, so we can help you choose the optimal media for your application. For all organisms in the portal, and just to complement our experimental data, we have developed a software tool, the Genome Spot algorithm, that uses amino acid composition of the genome to predict likely growth conditions. This model has been trained on more than 11,000 organisms across 4 different conditions: oxygen tolerance, pH, salinity, and temperature. The predictions suggest an optimum, a range, and we also provide confidence levels for these predictions. Next, we screen for functional DNA parts, specifically for plasmid origins of replication, ORIs, and resistance markers. We have developed a high-throughput screening protocol where we screen for 22 different origins of replication and 6 different antibiotic markers that are delivered via conjugation. Here we show the results of what worked and what didn't work for this organism. For example, you can see all the ORIs that were functional in this organism. And for each functional ORI, like PTHT15, we provide the antibiotic marker that yielded the positive results in green, chloramphenicol and spectinomycin in this case, and in red, the markers that didn't work. And we also display the concentration for the antibiotics that we used. We also provide confidence levels for these origins of replication to be functional in the organisms that we screen. For all of the assays, the main goal is to show you what worked and what didn't work so that we can save you time. You can also get these sequences to use in your own research, and I will show you how at the end of the presentation. So as a summary of the optimal conditions that we identified for this organism, we can provide information to answer all of those initial questions and suggest an optimal media composition and the temperature at which you should grow your cultures, a DNA delivery protocol, an origin of replication, the antibiotic marker, and the antibiotic concentration for the selection. And we also have some extra information that we host, like a draft genome and a list of publications for this specific strain. So now we have collected all this type of information for the group of extremophiles, and here you can see the growth capabilities in a diverse set of media. In this heat map, we show the maximum optical density reached in the cultures for the different organisms in each media formulation. The yellow boxes convey low growth, whereas the red boxes convey high biomass achieved. And as expected, the alkaliphiles thrive in alkaliphilic media with high pH, whereas the halophiles thrive in halophilic media containing high concentrations of salts. But the interesting thing is that in both cases, we were able to use more common media with lower salts and lower pH to support their growth. And this simplifies a lot their handling in the lab. What we can also observe is that the radiation-resistant organisms grow better in slightly basified media with a pH of 8 than the common formulations with a pH 7, which is the suggested formulation for these organisms. We can also look at the success of delivery of functional parts. And in this heat map, we show the origins of replication that we identified as green boxes for the different organisms. For example, if we take the organism example we discussed before, Halocalibacterium halodurans, we see that RSF1010 and these other 5 origins of replication are functional in this organism. And I would like to emphasize in here that this is the first step to make these organisms genetically tractable. Some of the key takeaways for this set of extremophiles is that we were able to suggest more useful media than what the depositors initially submitted to ATCC, which is very interesting. For some organisms, we identified a more productive media that yields to more biomass compared to the suggested media. For some organisms, we identified a lower lag time and thus a faster growth. We also identified a more accessible media, meaning a less complex media or more common formulation available in any microbiology lab for some of the organisms. And finally, we identified for one organism a media formulation that enabled the growth both in liquid and solid formats. As I mentioned before, extremophiles are not easy to work with, and we encounter some challenges. The first one being aggregation. Aggregation can make the growth parameter analysis very inaccurate, and also the presence of aggregates may result in inefficient genetic modification. The potential solutions that we suggest to overcome this issue include the use of alternative media and disrupt the aggregates, either by filtering, bead-beading, or sonication. The second challenge is slow growth. Long incubation times are usually coupled to low biomass, and this makes very difficult the genetic modification purpose. The potential solution involves using alternative media or using non-OD measurements to monitor the growth, like imaging. Finally, the third challenge that we encountered was identifying a single viable media. This is only a problem that has a bigger effect on conjugation, as the DNA delivery via conjugation requires that both the donor and the recipient are able to survive in the same media. And the most common donor strains are not viable in extremophilic media. So a potential solution is the use of metabolic models to identify media compatible with the conjugal host. Overall, across our 4 questions, we have generated experimental information on growth profiles, delivery protocols, DNA parts, and general information on the organisms that is available through our portal. Cultivarium has also made many tools and pipelines in-house, and 4 of these tools are available for you to use immediately in your lab. 2 molecular tools for identification of genetic parts, the POSUM toolkit for the identification of origins of replication and antibiotic markers. And this is the toolkit that we use to screen for DNA parts that is available in our portal. And the second one is Macro to identify ribosome binding sites and promoters. And these 2 kits are distributed through Agin and can be found in the link on this slide. There are also 2 computational tools, Genome Spot to predict the growth conditions, which is the computational tool that we use to generate the predictions that we display in the organisms page. And the second, Microbes for the identification of methylation patterns to avoid the restriction modification systems. On this link, you will also find the literature that we have released about the assays behind all the data that we have presented here today. So let's work together. We are excited and open to collaborate. Some of the many ways how we can support your work include that you can see and share the organism information on our portal. You can nominate your favorite organism for us to develop the genetic tools, and you can co-develop your application with us. If you want to learn more about Cultivarium, please scan the QR code at the top of this slide. And you can also request access to the portal, scanning the QR code at the bottom of this slide. Thank you, and I will now take questions from the audience.
Thanks, Dr. Moldis, for the excellent presentation. In just a few moments, we will begin our Q&A session. Please use the chat function available in the webinar program to submit your questions. And remember, the webinar presentation will be available on demand at www.atcc.org and on the Cultivarium website at www.cultivarium.com. We've had several questions come in, so let's go ahead and get started with the first one. The first question is, how many organisms has Cultivarium worked with?
So thank you very much for the question. And we have currently worked with more than 130 strains from 130 species in the lab.
All right. Let's see. The second question, what application spaces do you work on? How do you choose your organism?
So we work with a list of different applications, from organisms that would be useful for bioproduction to organisms that are interesting for soil technology, agriculture, and crops, and also organisms that are relevant for human health, like organisms from the human microbiome.
Okay. Let's see. Another question is, do you offer access to the portal?
That is a great question. And yes, we do offer access to the portal, and we love to have users from all kinds of origins. So I will encourage anyone to submit their request through to get the access to the portal through the link that I showed in the last slide. And you can also find this link on our website, cultivarium.org. And I just want to share that we have a brief vetting process for biosecurity reasons. Understood.
Great. Okay. Here's another question. Besides the ORI and the markers, do you test different promoters to drive the expression of selection markers?
Yes, we do.
Okay. The next question is, the 255 screen plasmids are 255 different plasmids, or the possum plasmid screened 255 times? And probably botched that question a little bit, but maybe you could understand the question.
Yeah. So the number of combinations that we show in that slide is the number of plasmid and organisms tested altogether. In fact, we have 138 different combinations of plasmids in that toolkit.
Okay. Here's another question. What is the OD600 normalized to? Is it meant to be OD600 of a 1 centimeter path length?
We use 96 well plates in plate readers. So the normalization is done to the no cell control.
Great. Okay, here's the next question. It says, great presentation. How frequently is the platform information updated?
Great question. The platform is continuously updated and as we test more and more organisms.
Okay.
And new data is uploaded also every week.
Okay. The next question is this database specifically for bacteria? Will it include fungi and other microorganisms?
Yes, we are extending into fungal organisms as well, but we also have protists and archaea.
Okay. Well, here's one. What is the main goal of your collaboration with ATCC?
So the main goal for the collaboration with ATCC, is to identify and pick interesting organisms that can be scientifically and technology interesting so that we can make them genetically tractable. If anyone in the audience is interested in a particular organism or application, I really encourage everyone to reach out to us through the website and also the links that we have provided during the presentation.
Okay, great. Let's see. Here's another one. You mentioned during the presentation that there's extra information that you host in the portal. Can you clarify what that is?
Yeah, sure. So apart from all the experimental and computational data that I showed you during the webinar, we also host information about sequencing of the strain, methylation patterns for some organisms. We also have publications. And recently, we have released a feature that is called Lab Lore, where you can ask questions to the community about a specific organism. You can also add handling tips or a general how to work with this organism, what to do, what not to do. And the great thing is that you can also reply to other comments, so it's very interactive. And you can like what other people added and discover other researchers working in that space through their profiles.
Sounds very, very valuable. How about this? What are the biggest challenges you face? What are some capabilities that you are thinking about developing to help you accomplish this awesome vision of making more microbes editable?
So we are aware that each organism is unique, and we always try to adjust our toolkits to these unique organisms. So, for example, with aggregation, we always make an effort to make the toolkits general. but we also try to address specific needs, like if they aggregate, what else can we do, like filtering, sanitating, and so on.
Okay. Do you have standard promoters and RBS parts that you can use across multiple bacterial species?
That is a really good question. And yes, we have a library of parts that includes ORI, markers, ribosome binding sequences, and promoters that we can use across different species. And again, you can find these toolkits on AdGen.
Okay. Let's see. Here's another one that just came in. Can you work with organisms that can't grow an auger? In parentheses, they put liquid only.
Yes, we can definitely work, and we develop our pipelines with organisms that only grow in liquid and not just in agar.
Okay. What are the plans for the methylation pattern side of things? Are you incorporating that into your tests with the POSSUM toolkit?
Yes, we have a bioarchive about identifying the methylation patterns, that is the microbe mode, and I really recommend everyone to look it up. And the main thing is that we use it to help avoid the restriction modification systems. And we have a plan to develop these tools to avoid it.
Okay. More questions are coming in. Why did you decide to focus on extremophiles?
So as mentioned during the presentation, extremophiles are organisms that play a very important role in many, many processes. They produce industrially relevant chemicals, and even though they produce these molecules, they are very poorly characterized in terms of the genetics. And sometimes that makes it really difficult for industrial purposes for their adoption. And that's why we decided to initially focus on these organisms.
Okay. Here's another question. Does the search tool you showed only work on Cultivarium's data?
The advanced search tool is a natural language search, and it works with all the data that we host in the portal, and not just the data that we generate.
Okay. Here's another one. What other features are you planning on releasing for the portal?
So there's one tool that is going to be released very, very soon, and it's a literature search that returns a list of microbes related to a keyword that you input with a list of publications. And you can also find links to those publications. And the other thing that you can do with this tool is that you can group the results by different taxonomic levels.
Okay. Let's see. How can I provide feedback or ask questions about the portal or what Cultivarium works on?
So there is a Work With Us page on the Cultivarium's website, and it contains a link to contacting us via email. If you have questions or feedback about the portal, you can reach out to us via the pop-up widget at the bottom right of the portal queue. So these are the several ways that you can provide feedback.
Great. By the way, we're getting a lot of good, amazing presentation, really positive results from folks attending. Another question someone asked was, have you developed CRISPR-based activation protocols for activation of cryptic biosynthetic gene clusters in bacteria?
Yes, so we are constantly developing new tools, and you will have to wait for release soon. And it will be for both random and targeted editing.
Okay. Is there an API for the data?
It is on our roadmap.
Okay. Another question was, do you show negative results?
Yes. That is one of the big things that we want to do, and our data tables in the portal display both the positive and negative results, so you can see what it worked for us and what didn't work, so that we can save time for your experiments.
Great. Okay. Well, I think that, oh wait, maybe, oh, here's another one that just came in. Have you considered characterizing strains or traits that are important for large-scale fermentation? In parentheses, they say, tolerant of oxygen and feedstock gradients, genetic stability over long seed trains, limited aggregation, etc. And I can read that question again if you need me to.
No, thank you. And the answer is yes, absolutely. We are very, very interested in traits that are relevant for fermentation. We are working on many of these right now because we think that many organisms can have potential to improve the biofermentation, but there's not enough information. So please get in touch with us because we are always looking for suggestions and ideas. Great.
Okay. I think we had some really great engagement today with the Q&A session. Like I said, there was a lot of kudos and amazing presentation comments. So I wanted to convey that to you, Dr. Moldes. So I want to thank everyone for attending this webinar. This concludes the webinar. Thank you, Dr. Moldes, for your time and your expertise. If anyone would like to learn more about Cultivarium, please visit their website at www.cultivarium.org. And for more information about ATCC, navigate to www.atcc.org. And as I mentioned earlier, the webinars will be available on demand on these websites. Please reach out to us if you have any questions. Otherwise, Dr. Moldes, thank you again so much for your time. And thank you, everyone, and have an amazing day.
Thank you very much.